Welcome back. You are listening to a really special edition of The Deep Dive. Today we are presenting the OriGene Technologies podcast.
It is great to be here, and this really is a special one. We're looking at some absolutely cutting-edge science, but we're also going to pull back the curtain a little bit on how that science actually happens.
Right.
We're partnering with OriGene Technologies for this dive because, well, they're a huge part of the infrastructure of discovery. They provide the genomic tools, the antibodies, cDNA clones, all the reagents that let researchers actually see what's going on inside a cell.
And today, we're going to see exactly how those tools were put to work to unpack a mystery that's been keeping, I think, a lot of virologists up at night for the past year or so.
For sure.
We're digging into a major paper. It was published in Nature Communications in late 2025.
The title is "The Potential of H5N1 Viruses to Adapt to Bovine Cells Varies Throughout Evolution."
Which, let's be honest, it sounds a bit dry. But the story behind it is anything but. It's basically a detective story about H5N1 avian influenza bird flu and its sudden, completely unexpected jump into dairy cattle in the U.S. That was back in March 2024.
And unexpected is really the key word there. I mean, people forget how weird that was. Before 2024, if you asked any virologist about cows and the flu, they would just shrugged.
Right. It wasn't on the radar.
Not at all. Cattle just weren't seen as a major host for influenza A.
And then all of a sudden, it wasn't just on the radar. It was everywhere.
Exactly. This specific genotype, B3.13, it didn't just pop up in one cow here or there. It spread like wildfire across herds in over 17 states. It showed us the virus had found a brand new playground.
So the mission for this deep dive is to get at the why. Why did this specific version of the virus take off in cows when others didn't? Was it a fluke? And importantly, we're going to highlight how researchers used very specific tools, like OriGene's IFIT1 antibody, to actually visualize the immune system fighting back.
Precisely. And what I love about this study is that they didn't just look at the outbreak virus, they looked at the whole history of the virus. The researchers basically built themselves a time machine.
Okay, I want to get into that time machine idea, because the experimental design is so often where the real genius is, right? How do you test 60 years of viral evolution in a lab without, well, waiting 60 years?
You build a library. The research team, they were led by scientists at the University of Glasgow, and they created this massive panel of over 80 recombinant viruses.
80? Wow.
Yeah, and they use a technique called 2.6 reassortment.
Okay, hold on. 2.6 reassortment. That sounds a bit like a tax code. Let's break that down for everyone listening.
Okay, so think of a flu virus like a car. You've got the exterior, the body, the paint, the door handles, and that's what determines how it gets into a garage, or in this case, a cell.
Right.
Those are the surface proteins, hemagglutinin and neuraminidase.
The H and the N in H5N1.
Exactly. So in this experiment, they wanted to control the variables, so they kept the car body the same for all of them. They used the surface proteins from a standard lab strain called PR8. So on the outside, every single one of these 80 viruses looked identical.
So they all have the same key to get into the cell.
Yeah.
That takes entry out of the equation.
Right. But they swapped out the engine. They took the 6 internal gene segments, the machinery that actually replicates the virus once it's inside, the stuff that does all the work, and they sourced those engines from wild H5N1 viruses going back 60 years.
That is just a fascinating way to isolate the variable. It's like building Frankenstein viruses. So you have one with an engine from 1959, another from 2002, and then one from the 2024 cow outbreak, and they just race them.
In a way, yeah. They wanted to isolate the role of that internal machinery. See, if they had just used the wild viruses as is and one did better than another, they wouldn't know why.
Was it because it could enter the cell better? Or copy itself faster?
Exactly. By keeping the outside constant, the 2 genes, and swapping the inside the 6 genes, they knew that any difference in performance was purely down to that internal engine.
Okay, I'm with you. So we have our Frankenstein viruses. Now, where do they race them? Because, I mean, a standard plastic Petri dish doesn't exactly scream dairy cow.
No, it doesn't. And that's a huge limitation in a lot of virology. Cells in a dish, they just behave differently than cells in a body. So these researchers, they went a step further. They used bovine skin fibroblasts, which is, standard.
Okay.
But then they used ex vivo udder explants.
Other explants. I'm assuming that means
Yeah.
actual slices of tissue.
Yes, real tissue taken from the udder that's kept alive in culture. And this is crucial because it mimics the actual environment, the structure, the cell types, the temperature, everything the virus would face in a real dairy cow.
It's the difference between testing a car on a dyno versus testing it out on the actual road.
Exactly.
So let's get to the results. We know the 2024 virus works in cows because, we saw the outbreak. But did all the other bird flu viruses manage to replicate in those cow cells?
Not at all. And this is where that time machine idea gets really, really interesting. There was this huge variation in what they call fitness, or how well they could grow.
Okay.
So as you might guess, the viruses that were actually taken from the outbreak, the B3.13 from cows, another one from goats called B3.6, these were the gold medalists. They replicated the fastest, they reached the highest titers.
And titers just means the viral load, the amount of virus they produced.
Correct. High titers means it's making millions of copies of itself very efficiently.
Okay, so the cow virus is good at being a cow virus. No surprise there. But what about the older bird viruses? Those engines from the 90s or the early 2000s?
They were the clear losers. I mean, viruses from 1959 or 2002, before this modern 2.3.4.4b clade really took over, they just replicated so poorly in the bovine cells.
So this ability to hijack cow cells, it isn't something H5N1 always had. It's a new trick.
It's a relatively new feature, yeah. But were there any surprises, any dark horses in this race?
That's what I was going to ask.
Yes. And honestly, this is the part of the paper that should really make us all sit up and pay attention. There were some intermediate winners, specifically a genotype called EA2022BB.
Catchy name.
I know, right? But here's the thing. This is a virus that's been found in wild birds and in some fur farms in Europe. It has not caused a massive cattle outbreak yet.
Yeah.
But in the lab, in those udder explants, it replicated really well.
Wait, so you're telling me there are bird flu viruses circulating in Europe right now that are essentially preloaded with the ability to infect cows?
That is exactly the implication. It suggests that the U.S. outbreak wasn't necessarily some 1 in a billion fluke mutation that just happened in one cow in Texas. The avian virus population has, as a whole, been evolving towards this capability. The engine was already getting tuned up in birds before it ever saw a pasture.
That is unsettling. It completely changes the narrative from bad luck in America to global vulnerability.
It does. It suggests the biological barrier between birds and cows is it's thinner than we thought.
Okay, so if the engine is better in these newer viruses, what exactly is different under the hood? What makes the B3.5 cow virus so special genetically?
To get that, we have to look at the polymerase complex. We talked about the engine. This is like the pistons. The polymerase is the photocopier of the virus, the machine that copies its genetic code.
And they looked at the PV2 gene. I feel like we always hear about PV2 whenever there's a flu scare.
You do, because it's a usual suspect for host adaptation. Birds run hot. Their body temp is around 40, 41 degrees Celsius. Mammals, like us and cows, were cooler, around 37.
Right.
A polymerase that's built for a hot bird often just seizes up in a cool mammal. It's like trying to start a car in the dead of winter.
So the virus needs a cold weather package.
Exactly. And they found a specific mutation, M631L. It acts like a turbocharger for replication in mammalian cells.
So is that it? Is M631L the turbo button? If a virus gets that mutation, is it game over?
Well, science is rarely that simple. It's definitely a major factor, but the researchers found that it's context dependent.
Eating.
Meaning you can't just slap that mutation into any old virus and make it a super spreader. When they put that mutation into some of the older avian viruses, it helped a bit. But it didn't turn them into the B3.33 cow virus.
Ah.
The B3.33 virus has a whole constellation of changes. It's not just one part, it's how that PB2 interacts with the nucleoprotein, the NP, and the non-structural protein, NS. The whole team has to work together.
It's like putting a Ferrari engine in a go-kart. If the transmission can't handle it, you're not going anywhere.
That's a great analogy. You need the whole package. And interestingly, they tested this polymerase in human cells too.
I was wondering about that. Cows are mammals. We are mammals.
Yeah.
Does this cow engine work well in us?
The activity was intermediate. It was better than a pure bird flu, for sure, but not as efficient as a fully human adapted flu strain. So while it's great at being a cow virus, it's not quite a human virus yet.
Yet being the key word there. But let's pivot for a second. We've talked about the attack, the viral engine. But the cow isn't just sitting there. The immune system has to be fighting back.
It is, and this brings us right to the battlefield of the interferon response. Interferon, or IFN, is the body's alarm system. When a cell gets infected, it pumps out interferon to tell its neighbors, hey, shut down the factories, lock the doors, there's an intruder.
And the researchers wanted to see if the cow virus was listening to that alarm.
Essentially, yeah. They treated bovine cells with interferon, basically pulling the fire alarm themselves, and then they tried to infect them. And they compared the cow virus, B3.13, against its avian ancestor.
And this is where we have to talk about visibility. Because you can't just look at a cell and know if the alarm's ringing. You need a way to see the molecular signals.
Right. This is where having robust tools becomes absolutely critical for getting clear data. The researchers used a technique called Western blotting. It lets you separate all the proteins in a cell by size and then use antibodies to find just one specific protein you're looking for. In this case, a protein called IFAT1.
IFAT1.
Okay, break that down. It stands for interferon-induced protein with tetratricopeptide repeats 1.
Let's just stick with IFT1.
Agreed. So IFA1 is a classic interferon-stimulated gene, or ISG. Basically, if the cell hears that interferon alarm, it starts cranking out IFIT1. If you see IFIT1 show up, you know the immune system is active.
And to see that IFIT1, they use a very specific tool from our partner today, OriGene.
They did. They used the IFIT1 mouse monoclonal antibody, and the clone ID is OTI3G8 from OriGene Technologies. And I want to pause on why that specific clone really matters. In Western blotting, specificity is everything.
Right.
If your antibody binds to the wrong thing, some random other protein, your data is garbage. It's just noise. And in a paper that's aiming for Nature Communications, noise gets you rejected. OriGene's TrueMab antibodies, they're raised against full-length proteins, which preserves the natural structure of the target. And that gives you these incredibly clean bands on the blot. It means when you see that mark, you know, for a fact, that it's IFIT1.
So using this OriGene antibody to see the battlefield, what did they find? This is figure 7 in the paper for anyone following along.
They saw something fascinating. The immune system was turning on. The IFIT1 band was there, lit up bright and clear thanks to the antibody. But the bovine B3.13 virus was effectively ignoring it.
Ignoring the defense.
It was just managing the response so much better than the avian version. The avian virus, EA2020C, it got crushed by the interferon response. When IFIT1 levels went up, avian virus replication just tanked. But the cow virus, it kept on replicating. It's developed ways to either dampen that signal or just power right through it.
So it's a double threat. It has the turbocharged engine we talked about, and it has stealth technology to ignore the immune alarm.
Exactly. And we only know that because tools like the OriGene IFIT1 antibody allowed the researchers to quantify that immune marker so precisely. If you can't measure the defense, you can't measure the virus's ability to overcome it.
Okay, so the virus can dodge the alarm system. But what about the bouncers? You mentioned earlier there are restriction factors, proteins that try to physically stop the virus.
Yes. The big one in cows is a protein called MX1. It attacks the viral nucleoprotein and stops it from working. And the study found that, yeah, bovine MX1 does restrict the H5N1 virus. It is fighting back.
So the cow isn't defenseless.
No. But the virus is in this constant evolutionary arms race, always trying to mutate to escape MX1. But here's where it gets really interesting and honestly a little scary when we connect this to humans.
I was waiting for the human connection. We've established the virus can handle cows. Does that help it handle us?
There's a restriction factor called BTN3A3.
BTN3A3.
It's a primate-specific antiviral gene. We have it. Cows do not. And normally, BTN3A3 is a major barrier protecting us from bird flu. It targets the avian nucleoprotein and just shuts it down. It's a big reason you don't catch the flu from a pigeon in the park.
Okay, so we have a shield that cows don't. That sounds like good news.
It should be.
Yeah.
But the researchers found that the bovine B3.13 virus escapes BTN3A3 restriction.
What? How? If cows don't even have that gene, why would the virus from a cow know how to dodge it?
That's the twist. It inherited specific mutations in its nucleoprotein, specifically at position 52 from its avian ancestors. And it just so happens that those mutations, which might have been totally random or adapted for some other purpose in birds, they also confer resistance to the human BTN3A3 barrier.
So it essentially picked the lock to the back door of human cells, completely by accident.
Basically. It didn't evolve this trait in the cow. It brought the trait to the cow.
That is, wow, that's a lot to process. Okay, let's take a step back and synthesize all this. We have a virus with a better engine, stealth capabilities against interferon, and it's already accidentally bypassed a major human defense. What does this all mean for you, the learner, listening right now?
It means we can't look at H5N1 as just a bird problem anymore. This study proves that the ability to replicate in cows is not a fluke. The 2.3.4.4b clade of viruses is, for lack of a better term, evolutionarily ready for mammals.
And it highlights the massive importance of surveillance.
Surveillance and deep molecular research. We wouldn't know about the interferon evasion without those Western blots. We wouldn't know about the protein levels without reliable antibodies. You can't fight what you don't understand.
Absolutely. And robust research needs robust tools, whether it's the H5N1 mouse monoclonal antibody from OriGene that we highlighted today, or their cDNA clones and other proteins. These are the building blocks of discovery. You don't publish high-impact papers in Nature Communications with fuzzy data. You need reagents that work.
It's the whole ecosystem of science. Industry provides the tools, academia provides the inquiry. And together, they solve the puzzle.
So we've covered the time machine experiment, the turbocharged polymerase, the invisible war with interferon, and the accidental lockpicking of human defenses. As we wrap up, what's the one thing you want the listener to walk away thinking about?
I want to leave you with a provocative thought about geography. We tend to think of the cow outbreak as a U.S. problem because that's where B3.13 took off. But remember that European virus we talked about? The EA 2022-BB.
The one from the fur farms that works so well in the cow tissue.
Exactly. That virus is circulating in Europe right now. It already has the internal machinery to replicate well in cows. So the question is, are we looking at a singular unlucky spillover event in the U.S.? Or is the global cattle population actually much more vulnerable than we realize?
And we just haven't looked hard enough in other places yet.
Precisely. If the machinery is already there and the wild birds flying over Europe or Asia, the risk isn't contained by borders. The gun might be loaded in more places than just Texas.
That is a sobering thought, but a really important one. We have to keep looking, and we have to keep testing.
And we have to keep trusting the data.
Well, on that note, we really want to thank the team at OriGene Technologies for partnering with us on this episode and for providing the tools that help researchers uncover these complex biological stories.
Indeed. It's fascinating work.
And thank you, the learner, for sticking with us through the polymerase complexes and the nucleoproteins. We hope you feel a little more informed about what's happening in the world of virology.
Always a pleasure to explore the data with you.
See you on the next deep dive.